In progressive inherited retinal diseases and advanced age-related macular degeneration, the clinical endpoint is known as no light perception, or NLP. This grim diagnosis represents the complete, irreversible biological destruction of the photoreceptor layer located in the outer retina. Clinically, this widespread cellular death manifests as a completely flatlined electroretinogram. The cells responsible for capturing incoming light simply no longer exist. Because the photoreceptors are gone, traditional restorative therapies designed to repair the retinal pigment epithelium offer absolutely no benefit at this stage. Likewise, gene augmentation therapies meant to preserve outer retinal function are entirely ineffective once that tissue has fully atrophied. You cannot preserve a structure that has already collapsed. For decades, the standard clinical response to an NLP diagnosis was to treat it as a terminal visual state, discharging the patient to palliative care and low vision support. But the eye itself is not completely dead. The damage is heavily isolated to those outer layers. Because the primary sensory tissue is irreparably destroyed, any future intervention must pivot away from repairing the biology and instead focus on sensory substitution. Retinal ganglion cells, or RGCs, constitute the surviving neural architecture of the inner retina and the primary target for bypass intervention. Look at this high-contrast cross-section of a diseased retina. There is a stark physical difference between the atrophied outer layers at the bottom and the structures closer to the front of the eye. When we strip away the dead and dying photoreceptor layers, a massive functional void is left behind. Yet, despite the total loss of the photoreceptors, the RGC layer remains robust and structurally intact. This remaining viable tissue allows for a concept known as a sensory bypass. Instead of trying to fix the damaged outer retina, clinicians can jump over it completely, interfacing directly with the surviving retinal ganglion cells. By artificially stimulating these RGCs, we can force them to transmit visual data payloads directly through the optic nerve and into the visual cortex. The long-term survival of the inner retina means that absolute blindness is no longer a terminal medical state. It is an engineering and biological interface problem. The first major attempt to achieve this sensory bypass in humans relied on physical epiretinal prostheses. The Argus II system, originally developed for patients with retinitis pigmentosa, served as the primary clinical proof of concept. The device functioned by placing a microelectrode array directly onto the retina to stimulate those surviving RGCs with tiny electrical impulses. This technical source image shows the implant's physical placement. The metallic microelectrode array sits directly on the atrophic macula. Notice the rigid, non-organic edges of the polymer array. It's a completely inflexible piece of hardware forced to interact with highly delicate biological tissue. The hardware did exactly what it was designed to do. It successfully stimulated the inner retina and improved functional vision in a subset of patients, proving the bypass theory was viable. While the bypass theory was sound, the actual physical implementation of the device presented severe clinical challenges. First-generation hardware proved the concept of RGC stimulation, but it simultaneously exposed the extreme mechanical limits of placing rigid devices on fragile, degenerating tissue. These mechanical limits became immediately apparent during the Argus II clinical trials for age-related macular degeneration. In a tiny cohort of just 5 AMD patients, there were 7 severe device-related adverse events. This OCT scan profile reveals the failure mechanism. Notice the physical gap between the rigid implant and the tissue below. At contact points, the implant edges press heavily into the remaining layers. This stress is severely exacerbated by the extreme fragility of atrophic AMD tissue. The clinical result of this friction was cataclysmic. The sheer mechanical stress caused the tissue to tear, leading to retinal detachment in most AMD patients. These insurmountable physical risks ultimately led to the Argus II being discontinued in 2019. Treating neurodegeneration with rigid hardware creates unsustainable physical volatility. To move forward, the field needed a complete shift away from mechanical implants. That shift arrived in the form of Optogenetics, a next-generation biological-technological hybrid designed specifically to solve the hardware shear problem. Instead of surgically inserting physical arrays, Clinicians use viral vectors to deliver gene therapy directly into the cells of the eye. These vectors carry a highly specific payload, genetic instructions that encode for light-sensitive channel rhodopsin proteins. Once delivered, these proteins begin to express on the surface membrane of the surviving inner retinal cells. This effectively rewires the tissue. Non-sensory inner cells are biologically forced to become primary photoreceptors, reacting to incoming light on their own. Optogenetics achieves the ultimate goal of sensory bypass without introducing a single piece of rigid hardware. The eye safely interfaces with light through its own biological evolution. This emerging technology directly challenges the pervasive clinical misconception that an NLP diagnosis warrants nothing more than basic low vision aids. The traditional mindset of discharging absolute blind patients from rigorous structural monitoring is entirely outdated. The modern role of the clinical optometrist or ophthalmologist is to act as a lifelong navigator for these patients. The new primary clinical objective for end-stage patients is maintaining a state known as trial-ready. Upcoming optogenetic and advanced cellular therapies have extremely strict anatomical prerequisites for trial entry. To qualify, the single most critical metric for future viability is the preservation of the retinal ganglion cell layer thickness. An NLP diagnosis is not a clinical endpoint. It is the starting line for an aggressive, lifelong campaign of structural retinal preservation. To keep a patient trial-ready, clinicians must actively identify and mitigate the primary ongoing threats to the inner retinal architecture. Conditions like untreated cystoid macular edema can actively degrade the surviving RGC layer over time, eroding the necessary anatomical foundation. Additionally, the progression of dense cataracts can physically block the specific light wavelengths required to trigger future optogenetic therapies. This necessitates a long-term clinical management strategy featuring continuous OCT imaging and structural mapping. We must track the anatomy to ensure aggressive edema management, even without visual acuity. It also requires comprehensive genetic testing to match patients with upcoming trial criteria. Removing cataracts and managing glare maintain the clear optical pathway required for emerging bypass technologies to reach the inner retina. By rigorously managing the physical architecture of absolute blindness, clinicians preserve the exact biological real estate required to eventually restore sight.